Welcome back. Today we begin a series of recordings dealing with brains connected to each other. You know that classically, neuroimaging studies a single brain doing a task or perhaps being involved in a resting state analysis, but humans are intrinsically very social creatures, and the real action perhaps takes place in the interaction of several brains with each other. The case can be made that a single brain in isolation is really quite dysfunctional. It has been shown that loneliness, a brain disconnected from other brains for extended periods of time, is a lethal condition leading to early mortality, perhaps 7 years of decreased lifespan related to the isolation and loneliness. So this then is a series of talks beginning with a paper by Talia Wheatley at Dartmouth College. She takes advantage of the fact that the business school on the college is all housed in one building. All students live together, eat together, study together, and basically form a very tight social network. So she asked the question, what does this mean in terms of the brain activity that encodes the social relationship among these students? Now, at this business school at Dartmouth, students in the business school need to fill out a questionnaire. And in the questionnaire, they describe who their friends is, who they go out with, basically forming the matrix for a social network. They also record a brief video stating where they're from, what they're all about, kind of like a Facebook introductory video. And so this data set lends itself easily to form a matrix to study the social network that these 270 or so students form with each other. And that is the starting point for Dr. Wheatley's study. So spontaneous neural encoding of social network position. And so the network science has recently emerged in psychiatry as a very relevant endeavor. There is a certain vocabulary that goes along with network science, which we will review here quickly. So nodes in the network indicate the students, and lines indicate the social reported ties between them. A subset of this network was then selected to undergo fMRI study to see if the network in some way is represented in brain activity. So here is then a presentation of the network. You see there's a bunch of blobs. Some are in gray and they are bigger than others. These are students which have a greater eigenvector connectivity. They are friends with other friends who are important to many other friends. So it's like a power network inside the regular network. You see, of course, there are other students in the periphery. So the eigenvector centrality is a very important measure of popularity and connectivity in the student population. You see some of the circles in orange. Now these are the students selected for the fMRI analysis. So we can then view the connection of these students in terms of degrees of separations from themselves, the social distance, and the extent to which they were well connected to other well-connected others, so-called eigenvector centrality, and the extent to which they are connected to otherwise unconnected individuals, called brokerage. Here's an explanation of some other terms in the next slide of clusters and networks. You see a hub in the center, There are edges and nodes. The nodes are connected by the edges to a hub. You have a cluster on the left-hand side, and you have an isolated network isolate on the left lower corner. Here's an example of what happens to a network when a central high eigenvector centrality person is eliminated. So this happened during the English Protestant uprising in England, and a certain John Bradford, who was quite central to the network, the large blob in the middle, was executed, and you can see that the network on the right-hand side unraveled after his death in a very significant way, highlighting the importance of people with high eigenvector centrality. Now, in terms of the power of social connectivity, here you see a bunch of primates, baboons namely, who groom each other and are interacting with each other. And again, the items of geodesic social distance are explained, namely the minimum number of intermediary social ties required to connect two individuals, and eigenvector centrality, a prestige-based centrality metric that considers not only how many connections a given individual has, but also the centralities characterized by each context. So high eigenvector centrality implies that an individual is well connected to well. In terms of the evolutionary antecedents and consequences of this, in primates, mean social group size increases with relative neocortex volume. So there is a log linear relationship between the group size that you can belong to and the cortical computing power that is required to compute the belongingness to such a network of increasing size. And in the next slide, you see a relationship here of the relative infant survival and composite sociality index. So the better you are connected as a female in this group, the greater the survival chances are of your offspring, pointing to a very direct relationship in terms of evolutionary selection of social connectivity. Now, getting back to the first paper by Dr. Wheatley, so here again is the network that she is working with and the folks selected for the fMRI study in orange. So to probe then the spontaneous encoding of social network position information, the group used representational similarity analysis, RSA, which distills from the fMRI data so-called representational dissimilarity matrices that indicate the degree to which particular brain regions distinguish between sets of stimuli or mental states. So this is a statistical powerhouse and analysis that we will not analyze in detail here, but we'll have you look at a video here in the next slide, and we insert the video here for your viewing pleasure as Dr. Wheatley explains the strategy that she has been using in analyzing the social connectivity of these 270 or so business students at her university.
I'm going to go through this really quickly because I don't want to spend a lot of time on it. I just wanted to illustrate a very simple point, is that we care about other brains. This is a study we published this year in Nature Human Behavior. This is Carolyn Parkinson again. And we took a cohort of TUC participants. We scanned them while they were passively watching people in their network. So for whatever reason, TUC students, when they arrive, have to get in front of a camera, and for 2 seconds they have to say, "Hello, my name is such and such, and I am from so-and-so place." No idea why, but it's like a video Facebook thing. So we have them lie in the scanner, and they watch a lot of these videos with the sound off. They see these people that they know moving around. And the upshot of this is we found that when people encounter, i.e. see, someone they know, they spontaneously activate all kinds of information about where that person sits in the larger social network that they share, right? So you can decode from the observer's brain where the person that they're observing sits in the larger social space. And we did that by using a searchlight to capture local neural response patterns, turn that into a local neural dissimilarity matrix, and we can see based on like distances away from each other in social space and also properties that these people have in terms of the position in their social network, like how central they are, which is how well connected they are to well-connected others, how much they bridge disparate cliques in a network versus are constrained to certain sort of sectors of the space. And we can start to build models not only of the neural data, but also of the social network data. We can build a model that captures if they're seeing their friend, or a friend of a friend, or a friend of a friend of a friend, that social distance. We can build a model that captures the centrality of the person that they're viewing, how central, the high sort of social status, hubbiness of a person. And we can model something like network constraint or brokerage, how the person that you're watching, whether they sort of bridge disparate cliques in a network, whether they kind of interface with different kinds of groups. And I'm not going to go into.
Okay, welcome back. Now you have reviewed the video by Dr. Wheatley, and you have a rough understanding as to what she is doing. So in the next slide, and as you can see, the technique is to show a brief series of images where the proband encounters either somebody close in the social network or more distantly related in the social network. And then the computer gets to work and analyzes the fMRI signals in terms of dissimilarity matrices, allowing you to assess the state of the brain and in relationship to the social connectedness measures. And as you can see here, the eigenvector centrality in orange invokes certain brain regions as very important and are predictive, in fact, of the relationship among the students to each other. So the intensity of the signal in eigenvector centrality plotted here on the surface of the brain allows you to predict who the next closest friend is in this proband's social network. There is also a distance, of course, which is the geodesic distance, and constraint characterizes those folks which are able to connect more distantly related subgroups to themselves. And you can see that different areas of the brain are invoked in producing those connections. So this then is a more, from the surface, 3,000 feet high approach to looking at social connectivity. The upshot of this study then is that social network proximity is significantly associated with neural responses in neural response similarity, in fact, in brain regions involved in attentional allocation, narrative interpretation, and affective responding. And they are exceptionally similar in how they attend to interpret and emotionally react to their surrounding. Now this then is the starting point for the next paper. And in the next study, Dr. Wheatley attempted to analyze whether brains of close friends are alike. Brains of similar feather joined together in a flock, of course, as the old saying goes. This question now is, do brains that are similar in terms of their responsiveness to stimuli also associating with each other? Here then again is her network. We already met it in the first paper that she is using for this study as well. And in this slide, she outlines the approach, namely, during an fMRI study, each subject watched a collection of video clips. These videos presented in the fMRI study covered a range of topics and genres, comedy clips, documentaries, debates, and so on. And they were selected so that the subjects were not familiar with these video clips, to make sure that they paid attention and to minimize mind wandering, and to evoke meaningful variability in responses across subjects, because different subjects may attend to different aspects of these clips, have different emotional reactions to them, and interpret the content differently, for example. Now I will now show you another video clip in which Dr. Wheatley explains the procedure of this protocol, and I'll see you back on the other side of the video.
It was very easy to find someone to replace them.
The water squeezes out of the cloth, and then because of the surface tension of the water, it's
insulting when a coach is making 5 to 10 to 15 times more than a college president.
These babies are not strong enough to cling, and they fall to the ground repeatedly.
It's at times like that you don't often think of funny things to say, but I did. And I turned to the rescue workers and I said, talk about a rough day at work. That was referring to spending the time up there being pretty rough. Talk about a rough day at work.
Okay. All right. So there was slapstick, there was more sort of mockumentary style humor and politics and science and all sorts of things. So we took the brains and we segmented, reconstructed, and parcellated them into 80 anatomically defined ROIs through FreeSurfer. And basically, we just took the average time series out of each of these ROIs and looked at how they correlate across subjects pairwise through the entire data set. So, so just a graphic of that.
squeezes out of the cloth, and then because of the surface tension of the water, it's
Okay.
Welcome back. You have now watched Dr. Wheatley present the study here, looking at how brains are similar among friends, which is the major finding. And here you can see what happens. You can expose these folks to different video clips and analyze their fMRI activity in various brain regions. The brain's been parcellated into 80 different areas which have been analyzed, and you can compute the correlations not only among the brain regions but among friends, or those that are more distantly related in the friendship hierarchy. So that is the basic technique. And here is one of the results. It's actually quite overwhelming. You see there is great similarity in the top row of various brain areas in terms of responsiveness and correlation, in terms of the similarity. And then as you go down to the second generation of friends, friend of friends and friend of friends, you can see that the similarities rapidly decline, indicating that first order friends or close friends have much in common in terms of their responsiveness to these video clips. And in the next slide, we see this expressed here as the average dyadic MRI response time series of similarities between these friends, overlaid on cortical surface model. And as you can see, the social distance 1, 2, and 3 shows a rapidly declining similarity. On the left-hand side is social distance 1, which is friends, social distance 2 is friends of friends, and so on. And you can see that the warm red and orange colors rapidly change into cool blue, indicating that there is not much correlation going on in friends of friends and friends of friends of friends. So yes, the answer then seems to be that if you are a close friend and you guys watch the same videotape, your brain areas respond in a very similar way to various stimuli, whether they be documentaries, whether they be emotional clips, whether they will be historical information, etc. And the next slide now shows you the social distance plotted against the similarity matrix, in a sense. And you can see how rapidly the similarity goes down from friend to friend of friend and friend of friend of friend. Now, as a cautionary note, do we become friends with people who respond to the environment similarly? Or do we come to respond to the world similar to our friends? Although the results of this study suggest that friends have exceptionally similar neural responses to naturalistic stimuli, due to the study's cross-sectional nature, we cannot ascertain based on these results alone whether neural response to similarity is a cause or consequence of friendship. Now, my money would be on the causal relationship, namely that similar brains associate with similar brains in forming friendships. And of course, I'm sure that Dr. Wheatley will pursue this further in perhaps more longitudinal studies, and we look forward to those results. In further talks about connecting brains, we will talk about the healing nature of touch and how touch connection synchronizes brains, and we will look at the breakdown of social connectivity in patients with borderline personality disorder. In the meantime, thank you very much for your attention, and we'll see you soon again at another talk from Behavioral Health 2000.